Okay, now recording. All right, so I'm going to send this email out. I just hadn't had a chance to send it out yet, but for lecture. So we're going to finish the digestive system today. I am going to cover the digestive physiology of the small intestine and then the large intestine. I'm then going to email you a little learning activity for you to complete, and then I'm gonna address any questions you may have about the um, digestion that occurs in the small intestine on Wednesday. Um, then on Wednesday, there'll be a quiz. It'll be a quiz for mastery, just like all of the other quizzes. Well, the last couple of quizzes that you've had, where you can take them over and over again, and take your highest score. Okay. That's going to open on Wednesday at the end of class, and it's going to close on Friday. Now, we're going to start the respiratory system, but I'm about a day behind, and I don't want to have a crunch for time next week when we're supposed to be finishing the urinary system. So I am going to place two relatively short screencasts on the introductory material on the respiratory system. It's not very difficult material. It's not something that I feel that I have to lecture on. But the stuff that comes after that, I think, is more important, that I'm actually lecturing on that in real time. So there's going to be a relatively short screencast. I think it's 15 minutes. I completed it this morning on introduction to the respiratory system and the upper respiratory system. Okay, and then there's going to be another one on the lower respiratory system. Again, it's not very difficult information. There's a lot of overlap with what you're doing in lab this week. Together, they're going to cover about 30, probably take about 30 minutes, okay, for you to watch them. Do read the material, and I say this in the screencast, read the relevant pages in your textbook before you watch the screencast. Treat them just like just like you were going to lecture, okay? And then on Wednesday, I'm going to start lecturing on starting with slide 40. So you don't have to wonder where the screencast going to cover and where is he going to start. I'm going to start on slide 40, okay? Which means that by Wednesday, you should have read most of the respiratory system. Certainly, I would say through up to control of respiration. So by Wednesday, read in your textbook up to control of respiration. Then you would have read in your book everything that I'm going to cover. Okay. All of the recordings from all the previous recordings of our meetings, of our lectures, have been placed on Blackboard through last Fridays. I want to remind you, your take-home exam is due on Friday at midnight. So make sure that is in Friday at midnight. Okay. For lab, last week we covered functional anatomy of the digestive system. You're going to have a quiz on the functional anatomy of the digestive system. It's due Friday at midnight. Your lab this week is respiratory system anatomy. The pre-lab for that is due Friday at midnight. Obviously, you have a lot due Friday at midnight, so don't wait till the last minute to complete those items. With the respiratory system anatomy, you should watch the presentation on Blackboard, and then do the pre-lab, watch the presentation on Blackboard, and then use the Quizlets to study and learn the material. You're going to have a practical next week, and the practical will be available today, just like all the previous practicals. It's going to open Thursday the 14th, and it's going to close Friday the 15th at midnight. Okay, and that practical is going to cover heart and blood vessels, digestive system anatomy, anatomy of the respiratory system. It's going to be just like the previous one. It's going to 50 questions, 42 minutes for you to complete them. You'll be taking the questions one at a time. And the Quizlets will not be available while the practical is open. So Even if you choose to take the practical on Friday, you won't have access to the Quizlets as of 8 a.m. on Thursday. Okay, so I'll get this email out to you shortly after our lecture today. Okay, any questions about any of that before we go on to content? Okay, so there's a question about the final. Bear with me just a second. Okay, so the final exam, there's a question of when the final exam is going to be. The final exam is going to be, per your syllabus, it's going to open Wednesday, May 20th at 8 a.m. It's going to close Friday, May 22nd at midnight. Okay, it's a couple of weeks away, so I'll talk more about that final as we get as we approach it. But while we're talking, there's going to be it's going to be two portions. One portion is going to be just like all your previous exams, and it's going to cover the new information, which will be digestive system, respiratory system, urinary system. And then there's going to be 50 multiple choice questions that will cover all of the other chapters that we've covered. Okay, and each one's going to be 100 points. Okay. All righty. So what we're going to do now is start covering digestive physiology of the small intestine. And then we're going to cover the large intestine. Okay, let me just make sure everybody's seeing this. Okay, all right, cool. So today we are going to cover the digestive physiology of the small intestine. Of all of the topics that I'm covering with respect to the digestive system, this is the most important because this is where most chemical digestion occurs and almost all absorption occurs. Here are our learning objectives. We're going to explain the following processes: digestion and absorption of carbohydrate, digestion and absorption of protein, digestion and absorption of lipids, and absorption of minerals, water, and vitamins. And then we're going to describe the regulation of the activity of the small intestine. So in other words, how does the body control this digestion and absorption of all of these nutrients? Okay, now before we start talking about how the body digests and absorbs organic molecules such as carbohydrates, proteins, and lipids, you need to know what they are. So hopefully, if you have forgotten what's a carbohydrate, what's a protein, what's a lipid, you have gone and reviewed those from chapter 2, chemistry. If you have not, or if you have forgotten these, it's going to be a little difficult to really grasp and understand the significance of the digestion and absorption of these molecules. Okay, so our approach in studying the digestive system has been to basically follow the pathway of food as it moves through the digestive tract. And as you study the digestive system, that is how you should study it. So go back to the, I can get it to start. Yes, go back to the oral cavity where food is ingested or brought into the mouth, right? And there is very little chemical digestion. It's mainly just mechanical digestion, breaking that food down into a bolus so it can then be swallowed. Although there is a little chemical digestion in the form of carbohydrate amylose being digested to a disaccharide maltose. That bolus is then swallowed and it enters the stomach. And you learn hopefully over the weekend that the stomach, there's not a whole lot of chemical digestion that occurs there. Probably before you watched the screencast, you probably thought, oh, that stomach acid is breaking down the molecules and digesting it, where in actuality there's very little chemical digestion that occurs in the stomach. The stomach basically is a storage unit for the food that you consume. There's a lot of mechanical digestion that occurs as that bolus is broken down to chyme, which is like a heavy cream consistency, and that chyme is then delivered slowly to the small intestine. But there's very little chemical digestion that occurs. Most of it is mechanical. And that, so what about that stomach acid? What was its function? Well, it's not so much to digest, but to prevent the proliferation of bacteria. Because think of the stomach. If you're storing food there and it's moist and it's warm and it's dark, that is like the perfect conditions for bacteria to thrive. And so what the stomach acid does is it brings the pH down so low that bacteria really can't survive, or they certainly can't thrive very well. So that's really the function of the stomach acid there. It's not so much digestion. Is there any chemical digestion that occurs in the stomach? Yeah, there's a little, in the form of proteins being digested down to polysaccharide. The polysaccharide called, I'm sorry, not polysaccharides, gosh, protein being digested down to a polypeptide, just a shorter chain of amino acids. And that's done by a enzyme called pepsin. And pepsin, if you watch the screencast, is actually what Pepsi-Cola was named after. I assume that they promoted Pepsi when they first came out with it as a digestive aid, and that's why they named it after that. So very little digestion and chemical digestion and almost no absorption occurs in the stomach. But that chyme, that heavy cream consistency, is then delivered to the small intestine. And it is in the small intestine that most chemical digestion and almost all absorption occurs, right? And so that's where we're going to focus the next hour or so of our time. Okay? So we are in the small intestine. As I mentioned, there's very little chemical digestion that occurs prior to the small intestine. Most carbohydrate and protein digestion, therefore, is going to occur in the small intestine. Right? Yep. If you could please mute your mic, please. Please mute your mic if your mic's not muted. Most carbohydrate and protein digestion occurs in the small intestine. A very small amount of digestion of carbohydrate has occurred in the mouth in the form of amylase, digesting amylose down to maltose, but that's it. A little protein digestion has occurred in the stomach in the terms of pepsin, breaking protein down to polypeptides. But notice that there's been no lipid digestion at all. Lipid digestion exclusively occurs in the small intestine. So, in summary, very little carbohydrate digestion occurs prior to the small intestine. Very little protein digestion occurs prior to the small intestine. Protein is broken down into polysaccharides, but that's about it. And almost no lipid digestion occurs. No lipid digestion occurs prior to the small intestine. Okay, so what that means is most digestion is going to occur, most chemical digestion is therefore going to occur in the small intestine. That's why the small intestine, what the digestive physiology of the small intestine is so important, and why I am spending the next 30, 45 minutes covering that. Now, when we talk about digestion, if you remember, is the breakdown of large molecules to smaller molecules. In chemical digestion, we're talking about the breaking of bonds. Do you remember those hydrolytic reactions we talked about at the very beginning of the semester, where water comes in, and we call hydrolysis reaction? Water comes in and breaks bonds. Those hydrolytic reactions are how carbohydrates and proteins and lipids are broken down. So you have a larger molecule, and then water comes in, and breaks that larger molecule down into smaller molecules. That's how polysaccharides are digested to di and monosaccharides. That's how proteins are digested down into polysaccharides and amino acids. Okay, let's first talk about carbohydrate digestion. Okay, now, so why is digestion even necessary? Well, ultimately, we want to bring nutrients into the body, and most nutrients are too big to be absorbed into the blood or into the into lymph, and so they have to be broken down. In terms of carbohydrates, the only carbohydrates that are small enough to be absorbed are monosaccharides. Anything bigger than a monosaccharide, therefore, has to be digested, broken down. So let's say you consume starch, like breads and crackers or pasta. You're consuming a polysaccharide called amylose. So you have amylose in the mouth. And what happens to that amylose? Well, there is some amylase in the saliva that will break that amylose down to maltose. Maltose is saccharide. It's sweet to the taste. That's why if you place a cracker in your mouth and you hold it there for a period of time, it'll begin to taste sweet. That's because that amylase is breaking that amylose down into maltose, and maltose is sweet to the taste. Now, is all amylose broken down in the oral cavity? No, some amylose does make it into the stomach where nothing happened to it, and then it therefore enters the small intestine. And in the small intestine, any amylose that wasn't broken down to maltose is broken down by amylase from the pancreas. So both the salivary glands and the pancreas produce amylase. So any amylose that enters the small intestine is broken down into maltose by amylase from the pancreas. What about other disaccharides, right? What if you consume sucrose or you consume lactose in milk? Does anything happen to it in the oral cavity? No. Does anything happen to it in the stomach? No. It enters the small intestine as sucrose and as lactose. All right. So we have disaccharides that you consume enter the small intestine intact as disaccharides. Well, I told you that only monosaccharides can be absorbed. So obviously there's something in the small intestine that can break down those disaccharides to monosaccharides. And indeed, that is the case. The absorptive cells, those simple columnar epithelial cells that line the small intestine, have those microvilli, which we call a brush border, that produce enzymes that can break those disaccharides down to monosaccharides that can then be absorbed. Now, I am not necessarily going to hold you to knowing the names of these enzymes. I've provided them here, right? Remember that enzymes almost always end in ASE, and they take on the names of their substrates. So sucrase breaks down sucrose. Lactase breaks down lactose. Maltase breaks down maltose. What I'm more interested in you knowing is that disaccharides are broken down into monosaccharides by enzymes from the brush border of these absorptive cells. Sometimes they're called brush border enzymes. Okay? So another nice sort of summary slide of what's occurring in the small intestine with respect to carbohydrate is shown on this slide that you have. So if any polysaccharide that's not digested in the mouth makes it into the stomach, nothing happens to it in the stomach. And then once it's in the small intestine, amylase from the pancreas breaks it down into the disaccharide maltose. And then any disaccharides that were consumed pass from the oral cavity through the stomach also into the small intestine, because there are no enzymes in the oral cavity or the stomach that can digest them. right? Once those disaccharides are in the small intestine, they are broken down by brush border enzymes to monosaccharides. Also, any monosaccharides that were consumed, right? Like let's say you consume glucose or you consume fructose, it passes into the small intestine untouched. It doesn't have to be digested. Once we have carbohydrates in the form of monosaccharides, now they're small enough to be absorbed. And there are protein carriers that allow them to be absorbed by the process of facilitated diffusion. Remember that? The carrier protein transports them into the absorptive cell. And then there's another carrier protein that transports them from the absorptive cell into the blood. All right. So when we talk about carbohydrates being absorbed, we're talking about movement from the lumen of the small intestine into the blood of the capillaries. Okay. So that is the digestion and absorption of carbohydrates. Let's now move to protein digestion. Now, I want to remind you that protein is composed of long chains of amino acids. Long chains of amino acids. Proteins are 50 amino acids long or longer. Do you think that a molecule that large can be absorbed into the blood? Well, the answer is no. So it has to be digested down, similarly to dye and polysaccharides, to molecules that are small enough to be absorbed. And so protein is digested down into amino acids, and you do have some absorption of dipeptides and tripeptides. That's just 2 and 3 amino acids. Okay, so let's talk now about how and where protein is digested down to amino acids and then how they are absorbed. So let's start with a protein, and basically this little round circle is going to be an amino acid. So a protein is a long polymer of amino acids. Are proteins digested in the oral cavity? Is there anything in saliva that digests protein? Well, the answer is no, right? So protein pretty much, when you eat a steak, that protein pretty much enters the stomach untouched, at least at the chemical level. Once that protein is in the stomach, what happens to it? Well, there is an enzyme called pepsin that's produced, and pepsin breaks protein down into polypeptides. In other words, that protein that's 50 or more amino acids long is simply broken down into shorter polymers. So we go from a large protein down to smaller polypeptides. But we're still talking about significantly large molecules that can't be absorbed. Okay? Those polypeptides then pass into the small intestine. In the small intestine, the pancreas produces enzymes called proteases, and those proteases break down those polypeptides into smaller polypeptides. Again, still too large to be absorbed. And so enzymes from the brush border of those absorptive cells, called peptidases, break those small polypeptides down to individual amino acids, or in some cases, some dipeptides and tripeptides. And now the molecules are small enough to be absorbed. Here's another summary slide showing this. So we have these large, so coming into the stomach, we had protein. Coming out of the stomach, we had large polypeptides. All right. Now, believe it or not, a few large polypeptides can actually be absorbed, right? They can actually be brought in by endocytosis, but that's not how most of your absorption occurs, all right? So don't worry so much about this route, right? Those polypeptides leaving the stomach are broken down by proteases from the pancreas to dipeptides and tripeptides. Some of those dipeptides and tripeptides can be absorbed, as shown here, right? Similarly to carbohydrates, facilitated diffusion into the will then diffusion into the blood. Okay, those di and tripeptides are broken down by peptidases into individual amino acids, and then those individual amino acids are transported into the absorptive cell and then into the blood. So we have a similar process as we saw with carbohydrates, where we have the pancreas playing a role at digesting the larger molecules into smaller molecules. Those smaller molecules still are too big to be absorbed for the most part, and so then the brush border enzymes are breaking them down to a small enough molecule that can then be absorbed into the blood. And like the carbohydrates, the products of protein digestion are absorbed from the small intestine into the blood. So when we talk about absorption of amino acids, it's from the small intestine into the blood. Okay, so we talked about carbohydrate digestion and absorption, protein digestion and absorption. Let's now talk about lipid digestion and absorption. And remember that no lipid digestion occurs in the oral cavity or in the stomach. It's exclusively digested in the small intestine. And lipids, if you hydrophobic, they don't mix with water very well. And most of what you consume is water-based, right? Think of all of the liquids you consume. They're mainly water with other substances in them. And a good percentage of most foods that we consume contain water. And so what will tend to happen is lipids will tend to clump up, just like it does if you try to mix oil and water. And so the body has to handle lipids a little differently than it does proteins and carbohydrates. So the liver, which is an accessory organ of the small intestine, secretes a substance called bile, and that bile is stored in the gallbladder if we're talking about between meals, and then it's secreted into the duodenum as needed, right? And what does bile do? Well, it emulsifies fat. Well, what does emulsify mean? Well, basically it mixes the fat with the rest of the contents of the small intestine, right? Sort of like if you've ever tried to mix oil and water, it'll all clump together by itself, but then you can stir it and it'll mix around briefly. Well, that's pretty much what bile does. It emulsifies that fat, right? So imagine you have this big glob of fat in the small intestine, because it's going to separate from the rest of the contents of the small intestine, right? Well, enzymes only act on the surface of that fat globule, so they're going to take forever to digest that fat. Sort of like if you were to take like a 2 by 2 by 2 cube of ice, the ice would only melt on the outside, so it would take like forever for that 2 by 2 by 2 foot cube of ice to melt. Well, how could you get that ice to melt faster? Well, you could crush it up, right? Increasing its surface area, and it would melt faster. So that's sort of what bile does. Bile comes in and then breaks that big fat globule down into smaller fat globules, and so now there's a greater amount of surface area for those enzymes to work, and so that allows the faster digestion of lipids, right? So that's the function of that bile, right? Just to physically break the bigger fat globules down to smaller fat globules for faster digestion. All right, so let's look at what happens. And I'll get back to your question, Michelle, at the end of this discussion on on digestion. All right, I haven't forgotten about you. So let's actually look at what is the nature of lipid digestion in the small intestine. Well, if you recall, most of what we consume in terms of lipids is triglycerides, right? Animal fat, butter, lard, parquet, canola oil, vegetable oil, corn oil, whatever. They're all triglycerides, right? So that's most of the lipid that we consume is triglycerides. Not the only, but it's most of what we consume. How is that digested? Well, nothing happens to it in the oral cavity. Nothing happens to it in the stomach. When it gets to the small intestine, there's an enzyme called lipase, right? Again, ACE lip lipase digests lipid. It's secreted by the pancreas. What does it do? Well, it basically takes off the first and third fatty acid. And what you have left is monoacylglycerol and 2 fatty acids, right? So that's the digestion of fat or lipid that occurs in the small intestine. All right, so what happens after that? So here we have this lipid droplet that's in the small intestine, okay? And in addition to triglycerides, we also consume some cholesterol. Right? If you're a meat eater, you consume some cholesterol because it's made by the liver of animals, right? So we also have cholesterol, but we don't digest cholesterol in the small intestine, right? Triglycerides, these big fat globules are emulsified to smaller fat droplets by bile. Then lipase breaks down those triglycerides into monoglycerides and fatty acids. And then those monoglycerides and cholesterol and those fatty acids are absorbed by diffusion into the absorptive cells. Now, you don't have to know this, but since it's included in the slide, believe it or not, after those monoacylglycerols and fatty acids are absorbed, the smooth endoplasmic reticulum reforms those triglycerides. So they're broken down to get into the cell, but then after they enter the cell, they're reformed into triglycerides, right? Cholesterol basically stays cholesterol. It doesn't really change. And then those triglycerides and cholesterol and some proteins are put together into this this unit called a chylomicron, which again you don't have to know, right? And then most of them pass not into the capillaries but into lacteals, which are lymphatic vessels. They are to the lymph system what capillaries are to the cardiovascular system, these small vessels that contain lymph. So unlike carbohydrates, unlike proteins, when we talk about lipid digestion, it's movement of lipid from the small intestine into the lymph system or the lymphatic system, and right, they are not absorbed into the blood. Okay, now I just want to be clear, right? The level of the level of detail which I covered here with respect to lipid digestion, you don't have to know. What I do want you to know is that triglycerides are digested by lipase from the pancreas to monoglyceride and fatty acids. And that's what's absorbed into the absorptive cell. And I want you to know that the products of lipid digestion are absorbed into the lacteals, not the capillaries. So lipid digestion is from the lumen of the small intestine into the lymphatic system or lymphatic vessels. Okay. Also, cholesterol is absorbed as is. It is not digested, right? It's absorbed from the lumen into the absorptive cell and then into the lymphatic system. All right, what about other substances? Well, what about minerals? Okay, now certainly different minerals are handled different ways, but it's far beyond getting into that level of detail in this class. Okay, what I want you to basically understand is other minerals like calcium and sodium and potassium and other minerals as well are absorbed all along the small intestine. Each has its own detailed process. Each involves different proteins, etc. Okay, just know that they are absorbed along the small intestine. Water. 90% of water that enters the small intestine is absorbed by osmosis. Fat-soluble vitamins, which are your A, D, E, and K, and the easy way to remember your fat-soluble vitamins is think ADDIC, right? That's how I remember them from college. ADDIC, A, D, E, and K. Those are your fat-soluble vitamins. Well, they're basically absorbed along with other lipids, right? In fact, anything that interferes with lipid absorption interferes with the absorption of those fat-soluble vitamins. But those fat vitamins are absorbed along the same process with the lipids. Water-soluble vitamins, with the exception of B12, are absorbed by simple diffusion all along the small intestine, right? Your water-soluble vitamins would basically be anything other than A, D, E, and K. So B1, B2, B6, right? All of those are water-soluble vitamins. Well, what about vitamin B12? Do you remember the intrinsic factor that was released in the stomach? Well, it binds to vitamin B12, and together they attach themselves to receptors in the small intestine, which then allow them to be absorbed by cell-mediated endocytosis. If you don't remember what cell-mediated endocytosis is, review Chapter 3, transport of substances across the plasma membrane. Okay, what about regulation of these processes? Well, as with all other aspects of regulation of the digestive system, the autonomic nervous system is going to play a role, and hormones produced by cells of the digestive system are going to play a role as well. The sympathetic nervous system decreases the activity of the small intestine. Well, that's not surprising because sympathetic nervous system is designed to prepare the body for increased activity. It increases blood flow to muscles and away from the digestive system. The sympathetic nervous system increases the activity of the digestive system, including the small intestine, through the vagus nerve. So parasympathetic nervous system activity increases the rate of digestion, rate of parasympathetic absorption. What about local hormones? Well, there is the substance called secretin, which is secreted by cells of the duodenum into the blood. Remember, it's a hormone. It does so in response to acid in chyme. So as chyme enters the duodenum, it drops the pH in the duodenum, and in response, secretin is released. Secretin targets the pancreas and causes the pancreas to release bicarbonate to neutralize the stomach acid, right? So the faster that, the more chyme that moves from the stomach into the duodenum, the more secretin that is released, the more bicarbonate is released by the pancreas. There's also another hormone that's released by cells in the intestine called cholecystokinin, all right? Cholecystokinin. It's released into the blood in response to protein and fat. It causes the release of bile, and it also causes the release of pancreatic juice containing lipase and proteases, okay, as well as amylase. Okay, so let's recap our learning objectives here. Explain the following processes: digestion and absorption of carbohydrate, digestion and absorption of protein, digestion and absorption of lipids, absorption of minerals, water, and vitamins, and lastly, describing the regulation of the activity of the small intestine, that is, the regulation of the digestion and absorption of these nutrients. Okay, so I want to take a pause here and address any questions you may have. And I know Michelle had a question. Let me pull it up. So, good question, Michelle. So the question is, how does lipid digestion absorption change if you've had your gallbladder removed? So for the most part, there's not much of an impact on a person that's had their gallbladder removed, because the liver continues to make bile and it continues to enter the duodenum to emulsify fats. However, the ability to store that bile has been greatly diminished. So where I do hear of people having some discomfort and some pains has to do when they eat a very high fat meal. My father, for example, had his gallbladder removed years ago, and he noticed that when he consumes like a salad where he uses vinegar and oil as the dressing, he will notice some pains and some discomforts. Right? To my knowledge, that's the only general negative impact of having your gallbladder removed. You have a decreased ability to store bile, and so sometimes if you eat a really high fat meal, there's not enough bile to emulsify the amount of fat that's there, and so there's some discomfort that occurs there. Can cause a little diarrhea as fat isn't digested and enters the stool. Okay. Other questions about that? Sure. Other questions about any of this that I have covered? So I'm going to email out to you, and I should have shown this before I stopped sharing my screen, but I'll go back to it real quick. Well, I'll go to this snipping tool. So I'm going to email to you this activity, and it says extra credit here. It will not be extra credit, just so you know. It's not extra credit. I'm not going to grade it. It's just something I suggest that you complete. Basically, what it's going to do is have you go over the material that I just lectured on. It's going to have you go to each of these nutrients and first identify different types of those nutrients, describe what happens to them before they reach the small intestine, and then list or describe what happens to them in the small intestine, what enzymes break them down, where those enzymes come from, what is the nature of their digestion, what are they before the enzyme breaks them down, what are they after, in what form are they absorbed into the body, and what does absorption mean? Does it go from small intestine to blood, or does it go from small intestine to the lymphatic system? Okay, basically this learning activity covers the learning objectives for digestive physiology of the small intestine. It's not something I'm going to take up. I'm going to go over. I'm going to ask if there are questions, and certainly if there are things you don't understand or something you're not sure about, write those questions down. And we'll go over them on Wednesday before we start the respiratory system. Okay, are there any questions? So I do not believe I have time to cover the large intestine. Let me take a look at the large intestine. Yeah, I've got 17 slides. The large and honestly, a good amount of the large intestine really around anatomy. So it's not critical that I lecture live on the large intestine. So I don't think that you will be negatively impacted, by watching the screencast on the large intestine outside of on your own. The thing that I just want to convey to you is that the large intestine is basically a dryer. What enters the large intestine is basically undigestible matter, what the body was unable to digest and therefore unable to absorb. It's about 50%. The dry matter is about 50% bacteria. And so what the body does, what the large intestine does, is absorb most of the water. It has to absorb enough water so that you don't dehydrate, but not too much water that it becomes difficult to move the feces through the large intestine. If it absorbs too much water, then you can become constipated. If it doesn't absorb enough water, then you can have a very loose stool or even diarrhea in some cases, right? So that's basically the function of the large intestine. Okay, and again, I feel very confident you can achieve the learning objectives of the large intestine on your own. And again, any questions that you have after you watch the screencast, bring them with you to our next session on Wednesday. Okay, so with that, the formal portion of this lecture, I'm going to bring that to an end. And if you have any questions on anything, it can be the topics we covered today. It can be other topics. It can be anything. I'm going to open the floor up